So today, I'm delighted to be able to introduce Dr. Helen Willsey, a truly outstanding young scientist and mentor who is an assistant professor in our department and a member of the Weill Institute for Neurosciences. I'll tell you just briefly about Helen so I don't take up too much time, but she began her academic career at Duke. She then went off to do a PhD at Yale, where I first became aware of her, which she was working in Dr. Jianshu's lab at Yale, working on mechanisms of developmental biology, molecular mechanisms, and where she won the prestigious Carolyn Slayman Prize in Genetics, which is really for the outstanding student and project in the Department of Genetics, of which I was a member. She then began her postdoctoral work at UC Berkeley, working with Dr. Richard Harlan, who is really one of the central figures in developmental biology, leveraging the Xenopus frog model system that you'll hear about today. And Helen went there to pursue really what was her idea, to try to use that system to understand, to leverage the emerging understanding of the genetics of autism to understand fundamental mechanisms in severe neurodevelopmental disorders. At Berkeley, she made a choice to come and take a look over on the other side of the bay, and I was thrilled when she elected to join my lab, although to say that she was a postdoc in my lab doesn't really capture the nature of the relationship. She came to the lab and really built an entirely new infrastructure within sort of the overarching structure of my laboratory. She started a Xenopus lab and pursued this idea that she brought to the lab to understand, to use her knowledge in developmental biology, human genetics, and psychiatric disorders to make fundamental progress in understanding molecular, cellular, and ultimately circuit level mechanisms. She sort of had a capstone for her training experience with a really spectacular paper in Neuron that not only, I think, clearly demonstrated the power of the approach, but opened up novel, truly novel insights into the biology of autism spectrum disorders, which we'll hear a bit about today. She's been an assistant professor now since 2022, and had a similarly steep trajectory. She's been enormously productive, both as a scientist and importantly as a mentor. I do have to say that while she was still in training, she won the Dean's Award for Excellence in Mentoring here at UCSF, a coveted and highly competitive award, as you know here. She also is a Chan Zuckerberg BioHub investigator, another highly coveted award. So today she's going to talk about her work at the intersection of developmental biology, human genetics, and psychiatry. I've heard Helen talk many times, as you can imagine, and delighted that she's going to be able to share that with us today. So thank you, Helen.
What a wonderful thing to be championed by Matthew State. Thank you for the kind introduction and for the many years of support and championing so many important causes. Thank you for framing the talk today in such a eloquent and impactful way. Yes, today I'm going to be telling you about our work studying autism genetics using frog models, which I promise will make sense in a minute. So all the work in my lab is a direct response to the explosion that's happened in the last 15 years in psychiatric disorder genetics. Most psychiatric disorders have a very large genetic component. And now, thanks to patient exome sequencing led by many people in this room, including Matt, have come up with many high confidence, large effect risk genes. This is even out of date. I think Belinda and Matt have new OCD genes coming out imminently. But I think that these genes are a really big opportunity to start to understand the molecular mechanisms of these disorders, taking down stigma against these disorders, and then also providing paths for rational therapeutics to be developed. This sequencing effort has been going on the longest, and we have the most genes known for autism spectrum disorder. So today I'll be focusing on those genes. So here's a snapshot of what the genetic architecture of autism right now looks like. And so you may have heard of SNPs or common variants from GWAS, genome-wide association studies. Those are down there at the bottom right, labeled in blue. Those are variations in the genome that are very common in the population. So they have a large allele frequency in the population. A lot of people have this variation, but individually, each one of those variants carries a very small risk. So those are not the genes and loci I'm going to talk about today. And so the ones I'm going to talk about are these rare variants of large effect. So if there is a mutation or a variant in one of these genes, it's very rare, so the allele frequency is low, but it carries over 100-fold risk for an autism diagnosis. So these genes are really the ones that provide the opportunity to understand what are they doing during brain development, what are their functions in cells, to start to understand the biology underlying these disorders. Now, you may even just be looking at this gene list, see that there's all sorts of different things. So where do you start, right? So there's a lot of challenges when you start with a gene list of 200 genes. So first of all, there's a large number of genes, over 200 now. How do you begin to think about those? If you do mouse models for 200 genes, that's just not going to be feasible or cost effective. A lot of these genes have unknown or very disparate functions. So it's hard just to look at the gene list and say, oh, that's what all these genes have in common. That might underlie some of the biology of neurodevelopmental disorders and autism. Then also there's just the complexity of the human brain, the protracted nature of brain development over embryonic development, and the sheer search space makes power analyses very challenging to be able to say, what do these genes do during brain development? And then really today I'm going to talk a lot about this idea of the problem of pleiotropy. And for me today, the definition of pleiotropy is going to be that one gene can do multiple different things depending on the context. And when you think about it, we have a limited number of genes, and the amount of cellular work that has to happen during development and then during homeostasis, it makes sense that multiple genes do many different things and wear different hats in different contexts. And that's a big problem in this field, because if you're trying to infer disease mechanisms just by looking at what we know currently about a gene, it may come up with some satisfying hypotheses, but may not be the most relevant to the disorder. And so with all of these challenges, I think we have a solution, which is frogs. And specifically, I'll tell you why. So this problem of pleiotropy is really tough. And so I wanted to think about frogs or a high throughput model organism such that I wasn't just studying one gene at a time. I could study 10, 20, 30 genes all in parallel to figure out what they have in common. Because you could imagine you study one gene, you see 6 different phenotypes, and you say, well, is that just like something that one gene does, or is that actually representative of what's going on for many different autism genes and therefore might be more relevant to the underlying biology? So to be able to study 10, 20, and now we've studied over 80 autism genes, I turned and learned the diploid frog model Xenopus tropicalis. So this is a great model because it has a simple diploid genome. Zebrafish have a duplicated genome, which makes genetics a little more challenging in sort of mapping the gene to the human gene, whereas frogs, it's very straightforward with high conservation. A single mating pair will lay over 4,000 embryos in a day. So that dish down there is actually a 15-centimeter dish, and each one of those little dots is a fertilized embryo that we can use for our experiments to understand gene function. They develop very rapidly. In only about a week, you have a free swimming, feeding, behaving, and I'll show you later, pooping tadpole to do your experiments with. Drug screening is really a dream, and I'll show you an example of that at the end, where you can just put the drug in the water. They're aquatic, and so you can just do 96 well plate format drug screening and see if you can modify a phenotype just by adding a drug. Okay, but if you remember one thing about frogs after today, please let it be this, which is that we can make half mutant tadpoles. So if we catch embryos right at that 2-cell stage, right after the first cell division of life, we can micro-inject CRISPR-Cas9 reagents to mutate any gene of interest we want. We co-inject a red dye, And 7 days later, you have a half mutant tadpole where only one side of the animal has your mutation of interest, and you can compare to the other side of the animal, having this within animal control to look at. It's quick, it's cheap, and because we have 4,000 embryos, this is usually what a typical day in the lab looks like, where we have thousands of half-edited animals where we can knock out whatever gene we're interested in, and today it's going to be autism risk genes, and then look at the animals and just say, what happened? This was the first gene I mutated, which was one that's required for eye development, eye pigment development. And you can see I injected it on the right side because now you have an albino or a pigmentless eye compared to the other side. And so then you can infer that this gene is required for eye pigmentation. So that's kind of how we wanted to go about it. During my postdoc, I developed various different imaging modalities to be able to make this high throughput and scalable, and so that we can look at all different kinds of cells in the brain, progenitors, excitatory, inhibitory, microglia, and watch them migrate and do all the things during development in a in vivo model. You can also take the brain out and image it in 3 dimensions by multiphoton microscopy, and you can rotate it around, and you can segment it, and you can measure volumes, and you can count the number of cells on your manipulated side compared to your unmanipulated side, and make precise measurements about what happened when you mutated your gene of interest. So at the start of my postdoc, when you would look at the list of autism risk genes, there were 2 clear categories that would pop out. The first is genes that have a known function at the synapse, right? So neuronal communication genes like neurexins and neuroligands and things that are really about neurons talking to each other. So that is that first bubble up there, and those are all sort of high confidence autism risk genes. And then there was another group of proteins that had been associated with autism that are known as chromatin regulators and transcription factors. So things that really affect gene expression. So they're in the nucleus, working to manipulate which genes are on and which genes are off. And so what we did during my postdoc was take the top 10 genes, I've circled them here, at the time by statistical association with autism risk. We knocked them down in this frog model, and we just said, what do we see? And I'm going to cut to the chase on this one because I want to get to some of the newer work. But one of the really surprising findings we found from this was that genes with known synaptic roles, like neurexin and SYNGAP1, I love SYNGAP1. It's so synaptic, it has synapse in the name. So synaptic RASGAP1. For both of these cases, when we mutated them, we actually saw structural differences in the brain. So now you're looking down on the head of the tadpole. And where that red arrow is the telencephalon region, or the forebrain of the frog. And there you can see when the neurexin is mutated on the right side, that forebrain region actually expands in size. And in contrast, when you mutate SYNGAP1 on the right side, that forebrain region actually contracts and you have a smaller forebrain there. This was unexpected. We thought that maybe this would happen with some of the chromatin regulators. People with variants in those genes often have micro and macrocephaly. But here, these synaptic proteins, if they really are only carrying out synaptic functions, should be acting on activity, not regulating the size of a brain. So this was our first clue that maybe we don't have a full picture of all the possible functions for these genes. Here are 2 more examples of ones that at the time were classified as synaptic, CATNL2 and DYRK1A. And again, for those 2, we saw a reduction in telencephalon size. And CATNL2 was one of the first ones discovered. And so when I wanted to kind of figure out why this brain was smaller, I started with these 2 genes. So when we raised antibodies against CATNL2 and just took a look, where is the protein during development? What you can see is that it's expressed very strongly in the brain, but then also the eye, the kidney, the olfactory epithelium. But what really struck us was that this CATNL2, which is the green, localized to a subcellular structure called cilia, which here are in pink. And don't worry if you've never heard of cilia, I will talk a lot about them. And by the end of today, you will know a lot about them. But it was really striking to us because not a lot of proteins localized to cilia, but there it was in these beautiful little puncta right along these hair-like structures that come out from the surface of cells. The next gene I studied was DYRK1A, and we actually thought this was going to be like a fun splicing project because at the time DYRK1A was annotated as a splicing factor kinase, and we did a bunch of deep RNA-seq and found basically no splicing changes. Anyway, we got some good antibodies, validated them, and looked to see where that one was, and lo and behold, it looked a lot like CATNL2, again showing up on the subcellular structure called cilia, again in that punctate pattern all along those hair-like structures. They weren't just there, they were also functional. We knocked down both of those genes, and you can see those pink cilia, hair-like structures go away, both when we CRISPR DYRK1A or when we CRISPR CATNL2. So they're both localizing to cilia, and they seem very important for the cilia to function. So let's pause for a moment and talk about cilia. So cilia are ancient subcellular structures that are present on almost all cells of the body. And they come in two flavors. One are motile cilia, which is on the left, which are the, much like the respiratory tract or the brain ventricles or the fallopian tubes, where a single cell, like here on the left, will put up about 200 of these hair-like microtubule-based structures, and they will actually physically beat to move mucus, cerebrospinal fluid, and other things in the body. They're also very important for heart development and congenital heart disease. Now, all other cells have the other flavor of cilia, which are primary cilia, which is a single, and it's that little pink antenna that comes off the cell. And they're really thought to be antenna. They take in signals from other cells, and then they transmit the information to the nucleus and even regulate neuronal excitability. They're really important for kidney development, neurogenesis, migration, which we'll talk about later. A lot of times the cilium is actually a thing that's guiding migration of a cell, neuron activity, and many key developmental signaling pathways, including those that set up the excitatory and inhibitory balance in the brain. But what a lot of people don't know is that actually a lot of really important neurotransmission receptors localize specifically to cilia on neurons. So this is an example from the hippocampus with somatostatin receptor 3 in green, and it localizes specifically to the primary cilium on those neurons. Similarly, MCR4, which mediates leptin signaling, is really important for satiety, and when this pathway is mutated, causes major obesity. MCR4 is localized specifically to the cilium. Even dopamine receptor D1 localizes specifically to the cilium and not to other places in the neuron. And there are also serotonin receptors like HTR6, serotonin receptor 6, that only localize to the primary cilium. And so I think there's a lot here to be discovered about the primary cilium on neurons, and the idea that there can be a lot of signaling happening in neurons outside of the synapse and at the cilium. Okay, just so happens that I was working in frogs, which is a great model for studying cilia. There's a bit of serendipity here. So this is what a tadpole looks like about 2 days into development. I like to show this off because people who study cilia, they're doing it in human cell culture, and to get this kind of ciliated epidermis, they have to wait 60 days in culture. So I get to show off and say 48 hours because I'm impatient. And so we can look and see these cilia. They are a great model for the respiratory tract. They're motile. You can see them beating. And so it's a very nice model system just to generally study ciliary biology. And so seeing both CATNL2 and DYRK1A, just the first two genes that we picked, having this key role in cilia, made us really step back, and we know about these signaling pathways in neurons, step back and say, what's going on with autism and ciliary biology? And when you actually look at all of the multi-organ presentations of a cilia defect, actually a lot of them co-occur with autism. So cilia defects are known to cause epilepsy, hydrocephalus, congenital heart defects, chronic respiratory problems, asthma, kidney issues, skeletal anomalies, vision loss, things like that. And so all these places where I have the red stars, there's clear comorbidity with autism. And so it really led us to this question of, do autism proteins more generally beyond CATNL2 and DYRK1A converge at the cilium? And so the graduate student who took this on is Alina Kostianovska, and she has a preprint out now that's under revision if you want to get all the details, but I'm just going to give you the overview. And so the first thing we did in collaboration with Belinda Wang was look and see if we take our list of high confidence autism genes and overlap it with proteomes from ciliary structures. So take all of the proteins that are in a cilium, take all the autism genes that we know and overlap it. Is there an enrichment of autism genes within those proteomes? And indeed, what you see, those blue lines are just the autism gene enrichments. And then the centriolar satellites, which feed cilia, and the neuronal progenitor centrosome, which is the base of the cilium, and then the neuronal centrosome, again, the base of the cilium. In all of those cases, autism genes are enriched for being in these ciliary proteomes, but not in an actin filament control proteome. This holds true even when you look at the all of the protein-protein interactors from these autism proteins that have recently been described. In fact, even in some cases, it looks like there's more enrichment. And even if you take away the baits and look at the prey only, it's still enriched, but never in the actin filament case. So this led us to say, okay, there might be something here. It looks like autism genes are present in these ciliary structures more often than you would expect by chance. And so then what we wanted to do was actually take a look and do a localization screen. So we took the top 30 of the autism proteins, the human version, added a green fluorescent protein tag on it, and then put it either into rat neurons or into our favorite frog skin, where the motile cilia are, and just said how many of these top 30, just by statistical association with autism risk, actually localize onto the cilia. And so we found 12 of those 13 that localize both to primary and motile cilia. As I showed you before, DIRC1A came out, that was a nice positive control, but we also got a bunch of proteins that were very interesting. So the first group of protein were these gene expression regulators, and specifically these chromatin regulators that are supposed to only be in the nucleus. But here, when we overexpress them in these cell types, we start to see chromatin regulators like CHD8 localizing onto cilia. Similarly, some of those synaptic proteins that have beautiful, well-characterized roles at the synapse, we also started to see at the cilium, including our favorite SYNGAP1, so synaptic, it's got synapse in the name, as well as some GABA transporters and snare proteins. So I'm going to give you a little preview of what that looks like when we do it in the frog skin at least. And so the green is the autism tagged synaptic protein, and that's one single cell, and the magenta are the cilia, and the bottom you can see the green channel alone. What's cool is that these all kind of have different localization patterns. For SYNGAP1, it really localizes to the base of the cilia, and then just the first part of the cilium that needs to be hyperstable for when it whips back and forth. STXBP1 is a snare binding protein that's actually localized right where targeted vesicle trafficking happens into the cilium. Just like the synapse, the cilium has a diffusion barrier. And so to localize dopamine receptor 1, somatostatin receptor 3, there has to be active delivery of those membrane receptors into the cilium that requires snare-mediated vesicle fusion events. And so it's very interesting to see a snare-associated protein right there where that happens. SHANK3 is a more scaffolding-like synaptic protein that also interacts with actin. And so actually you can see there in those cells around that middle cell, that looks very much like an actin network. And then it's also right there where actin interfaces with the base of the cilium. And then Norexin looks even different from that. It looks more like it's circling around the base of the cilium, providing stability on the exterior of it. So each one of these is going to be interesting to study and to go forward with, to think about all the different ways you could mess up a cilium and all the different ways that something could function at the cilium and more. So I'm just going to give you a little snippet now on the kinds of things we're doing to follow up on this. So for SYNGAP1, it's localizing there, but is it important there? And so we can use our frog model to mutate it on one side and compare the cilia on the mutated side to the control side. And when we mutate SYNGAP1, you see a loss of these cilia, a loss of those pink hair-like projections in 2 different manipulations. And what's nice in frogs is these cells are really huge for imaging. And so actually one thing we wanted to try was to see if we introduced patient variants into this protein, does that actually change its localization within the cilium? So this is the baseline localization where it's on the base of the cilium and then just a little bit up into the cilium. But if we add, if we mutate the protein to include a patient-derived missense variant, that middle one now causes the protein to go to the plasma membrane where it never was before, whereas that other mutation causes it to form aggregates inside the cell and pulls it away from the cilium. So we're excited about this. Now, this is all frogs. We wanted to see if we could translate some of this into human insights. So to do that, we borrowed from the primary ciliary dyskinesia field, a ciliopathy. So when in the nasal epithelium and the respiratory tract, when there is a cilia defect, those cilia can't beat. And so the beating of the cilia actually produces nitric oxide gas passively from the nasal passage. So when there's a cilia defect, there's much less nasal nitric oxide gas that comes out of the nose. And so this is actually used in primary ciliary dyskinesia as a diagnostic metric to be able to see a ciliopathy in a patient with respiratory issues. So we went out and we got a portable nasal nitric oxide machine. That's a 30-second non-invasive method. It's about the size of the iPad, so it was easy to bring it to the patient meetups. And importantly, a lot of these kids have pretty severe intellectual disability and behavioral challenges, and we've now done across a couple different genes, over 100 patients with this machine. And out of the 100, only 5 were unable to complete the test. So we like this because it's not invasive, it's 30 seconds, the affected individuals can complete it. And then we compare to an unaffected family member. So this is what nitric oxide looks like right now, comparing the unaffected family member, so a parent or a sibling, with an individual that has a pathogenic mutation variant in SYNGAP1. And so you can see it's human data, so it's messy, but there is a statistically significant difference in the amount of nasal nitric oxide coming out of the nose in affected individuals compared to their unaffected family members. So this really makes us think a lot about gene classifications and the idea that this is a synaptic protein, but we're seeing effects even in the respiratory system. And so that really made us come back and actually think about the idea that synapses and cilia are actually kind of very similar structures. They both have septins to make kind of a pocket. They're rich in cytoskeletal components. They're rich in receptors. They signal through calcium. And they're very important for sensing the environment, responding to the environment, and intercellular communication. Of course, cilia are the ancestral state. So it's an interesting idea to think about synapses perhaps evolving from cilia. Okay, so I've shown you these proteins that have well-described roles at the synapse now showing up and functioning at the cilium. But there's chromatin regulators, right? They're definitely in the nucleus, doing what we think they're supposed to be doing, right? Well, a couple of years ago, a postdoc in the lab, Michaela, was looking at the localization of these chromatin regulators and noticed something unexpected. So in that top there, that's a cell that's not dividing, it's hanging out. And when you look in to see where these autism proteins here labeled in green are, they're mostly in the nucleus, as you might imagine, for something that's regulating gene expression. However, then when the cell went through cell division and starts to create a mitotic spindle, a microtubule-based structure that then will separate the chromosomes, these chromatin regulators relocalize onto these microtubule-based structures. We did not expect that. And then, so we were a little skeptical of it, and we said, well, could you manipulate it? And so she took patient-derived variants in CHD2 to see if that changed its localization. And indeed, on the left, you'll see in mitosis, the reference sequence goes to the mitotic spindle. You add the patient variant, and now it actually sticks to the chromosomes and doesn't relocalize to the mitotic spindle. whereas that third variant, or the second variant, the one on the third column, does relocalize. And so this made us think that those localizations might actually be functional, and patient variants might actually disrupt these localizations. And much like cilia, the mitotic spindle is made up of microtubules, which is made of tubulin. And so tubulin is a big actor in the cell for all sorts of trafficking and delivering cargoes, but also cilia and spindles. So this made us rethink what these chromatin regulators might be doing. Okay, so let's back up for just a second. So chromatin regulators are usually in the nucleus, and what they're doing is they're adding tags onto histones. So the histones will wind up the DNA and package it, and chromatin regulators can come by and put an acetyl group or a methyl group and sort of change the conformation of the DNA and allow certain genes to be turned on or off, more or less. And so it'll regulate that packaging. What I didn't realize is that tubulin has a very similar thing going on, where it has these C-terminal tails on its dimers, and there's many different proteins that come on and add a acetyl group, methyl group, phospho group, and change the conformation of the tubulin such that it changes the stability of the structural components of the cell. And so there's actually a lot of parallels between the histone code and the tubulin code in thinking about post-translational modifications. And there's a lot of examples of chromatin regulators that act both on histones and on tubulin. And so again, it's this idea of the first thing we know about a protein is just the first thing we know about a protein. So these are chromatin regulators, they're in the nucleus, they're modifying histones. But when they're outside the nucleus, a lot of them are modifying tubulin and regulating microtubule stability during mitosis and potentially in the cilium. So what we wanted to do was look at some of those tags, the phospho, the acetyl, the methyl, in cilia, and see if that code, the tubulin code, is it alive and well on cilia? And indeed, we got specific antibodies to all the different post-translational modifications on cilia, and they have beautiful spatially separated patterns where the acetylated tubulin is the top 80%, but then at the base, it's polyglutamylated tubulin. And so there's these really intricate patterns of where along the cilium these post-translational modifications live. And so that really made us think that maybe these chromatin regulators really are on cilia doing things in the tubulin code to regulate microtubule stability, which is so important for cilia. Indeed, when Michaela looked with validated antibodies against these chromatin regulators like CHD2, CHD8, and POX-Z, shown here in green, yes, they localize onto cilia themselves. And then we went one step further and she knocked them down in frogs and cilia, and they actually have kind of different phenotypes. But in all cases, when you knock down these proteins, you have cilia defects in the frog epidermis. So this really now, all these red proteins I've shown you localize to the cilium. And so we're able to bring together these disparate functional annotations and actually see that they can meet at the cilium there. We also think that this convergence is probably an underestimation because, for example, here's one of the top 30 autism proteins, RFX3. It's black because it's not on cilia. It's a transcription factor, and so it is in the nucleus, regulating gene expression. But I can tell you RFX3 is one of the canonical transcriptional regulators of cilia formation. And so while this isn't localized to cilia, it definitely has a role in controlling cilia formation. And so we think that this is just the surface of what's going on with these proteins in cilia. Everything today I've shown you is rare variants. What about some of these more common syndromic forms of genetic autism? And there is literature out there to support the fact that even things like TSC1 and 2, the mTOR pathway, MECP2, Fragile X, CDKL5, all of those have been shown to cause cilia, either length or ciliation phenotypes in neurons. And earlier this week, actually including UCSF investigators, there was a study showing that the ciliation and cilia length is different in 16p11.2, shorter cilia in the deletion and longer cilia in the duplication. So we think this may go beyond these rare variant classes and include even other genes as well. Okay, so with the time I have left, what I want to talk to you about is, now that we have this insight that a lot of autism proteins function on cilia, what can we actually do for somebody once you have that kind of insight? And so for me, what we've been thinking a lot about are the off-disabling comorbidities that come along with autism, many of which are known to have a ciliary component. And so, for example, cilia are really important for kidney development. And so for one of these genes, DYRK1A, when we started studying it, it wasn't appreciated that individuals with DYRK1A variants would have any sort of congenital kidney anomalies. But because, and this was in collaboration with Rachel Miller, because when she knocked down DYRK1A and got cystic kidneys in the frog, and I showed that it's expressed in the developing kidney, and we now have this insight about the cilia, then when they recruited families and did ultrasounds and other patient surveys to look at congenital anomalies of the kidney and urogenital tract, then they found 11 out of the 15 patients indeed did have often agenesis of one of their kidneys. So again, understanding that this may be a multi-organ issue caused by a common mechanism allowed us to then make hypotheses that could inform patients. And so now folks with DYRK1A will often go and get an ultrasound of the kidneys to check that out. The other comorbidity we've been thinking a lot about is a well-established comorbidity between congenital heart disease and autism. So a diagnosis of either one of those increases the prevalence of the other one. And frogs are really nice to study that because you can just flip the tadpole over and take a look at the heart. And so we've done a lot of work in this space, some of it published just earlier this year with Nia as the first author. And actually what happened is we were screening congenital heart disease genes to see if they had any role in neurons. And actually the ones that did, we found a cluster that are really enriched for ciliary biology. So no matter how we're trying to come at this, we keep ending up at cilia. And so cilia are very well appreciated for their roles in congenital heart disease. And we think, again, that this comorbidity may have some sort of basis in shared biology at the cilium. Okay, and then the last thing I want to share with you is this idea of when something is at the cilium, oftentimes that can disrupt gut neuron development. And so this project was led by Kate McCluskey, a graduate student in the lab who wanted to tackle this problem. And she really wanted to know, why do autism and gastrointestinal issues co-occur? So it's well established that an autism diagnosis increases the frequency of gastrointestinal issues several fold. And so she wanted to use frog models to think about this. So that's a frog gut. And the yellow ones are the enteric neurons, so the neurons that actually innervate the gut and control gut motility. And now that we have all these high confidence autism risk genes, can we think about what these autism risk genes are doing during gut development that may inform what's going on? Because of course, there's a lot of ideas about why autism and gastrointestinal issues occur, including it being secondary to behavioral issues or microbiome issues. She wanted to take a genetics first approach and say, what are these genes doing? So first thing she did was look at patient survey data from Simon Searchlight. So these are cohorts of individuals, all with a variant in the same gene. And then just a % of those individuals that have GI symptoms. And so the affected individuals have a large prevalence of GI symptoms, whereas the unaffected family member, which would be gray bars, except you can't really see it because there's really very few unaffected family members that report GI issues. And so there's a high, there's a prevalence of GI issues in these cohorts, so that these genes could be helpful for understanding this co-occurrence with gastrointestinal symptoms. And then she was also able to look at medical record data for several of these genetically defined patient cohorts and see that actually the largest contribution to these GI issues is GI dysmotility. And so actually constipation and inability to move the gut. She then looked at the expression patterns of these autism genes in the developing human gut and found enrichment of the autism genes, both in those enteric neurons, the neurons that innervate the gut, as well as their migratory progenitors, which was really interesting to us and made us think that actually the development of those neurons may be affected by these mutations. The frog, again, comes through for us. Very easy to study the migration and formation of the enteric neurons in a frog. As you go through development from top to bottom, you start to see these little dots come in from the head into the gut. Those are the enteric neurons marked by POX2B. They come from vagal neural crest. They migrate in from the top of the animal. And so then this sets up a paradigm where we can actually use our cute half and half trick and say, when you mutate this gene, what happens to the enteric neurons? Do these genes actually play a role? And the reason we thought that this might work is because in the zebrafish literature and in other model organisms, it's well established that if there's a cilia problem, those neurons have trouble migrating into the gut. And so we tested that in Xenopus. This is a TTC8, or also known as BBS8, knockdown, where you can see that those neurons don't migrate nearly as far into the gut. And so we said, well, let's take proteins that we've seen at the cilium and knock them down. Do we see a similar thing? And yeah, so for 5 different autism-associated genes with disparate annotations, synaptic, chromatin, all that, you can see that compared to the control in the top left, where the neurons have migrated and colonized 3/4 of the gut, now when you lose those other genes, there's issues with those gut neurons migrating into the gut. I love a good use of frogs. So then we wanted to see if we could actually model the dysmotility that's seen in these individuals with DYRK1A variants. So could we actually develop methods to quantify gut motility? And so what Kate did was she fed these tadpoles food with fluorescent beads for 2 hours. You can see them uptake it in their bellies. You can watch it go around the, and they're just gonna poop. There, yeah. There's another one. Nothing like watching a tadpole poop. And so actually she could scale this up. She could put them at 20 animals per well for 3 hours and let them defecate. We lovingly call these baskets porta potties. And then you could remove the tadpoles, leaving the excreted beads behind, and then just count the number of beads left behind as a metric to understand, to quantify gut motility. And so we first did that DYRK1A gene because there's a lot of helpful tools to study DYRK1A, including a kinase inhibitor. And so this TG003 is a kinase inhibitor. And so the boxes on the bottom are just representative, false colored images of the beads left behind. So everywhere we see a black dot, that's a fluorescent bead left behind. In our control DMSO, you see the amount of beads that they secrete. But then when they're grown in the context of a DYRK1A loss of function, they don't excrete nearly as many beads. Because now this was in multi-well format, we said, actually Matt suggested, we should do a drug screen. As always, he is correct. So then we did a gut motility screen. And so we could then we took 39, I think, FDA approved drugs that have some sort of connection either with neurotransmission or with a known function in gut motility, like caffeine or antibiotics. And just on wild type tadpoles said, which of these drugs can increase gut motility and which of these drugs can prevent gut motility. So we have a number of hits and we arbitrarily had a cutoff of 1 standard deviation compared to the DMSO control, and a bunch of drugs that were able to cause the tadpoles to secrete the beads versus ones that had them hold on to it. And you'll see at the top, 3 of them are in the serotonin pathway. And one of the things we knew at the time was that in the Xenopus enteric neurons, our gene of interest, DYRK1A, localizes very specifically to the enteric neuron cilium, which I'll remind you is also exactly where serotonin receptor 6 localizes to do serotonin signaling. And so given all this together, what we then wanted to do was go back into our DYRK1A model where the tadpoles don't excrete as much, and now take some of these serotonin agonists and see if in that context they could move the gut. And so, yes, they could. The top is the GMSO control. The second one is this DYRK1A inhibitor. Like I showed you before, they poop less. And then we added the common SSRI Lexapro to these animals for an acute treatment of only 2 hours. And that was able to move the gut back to almost baseline amount of beads secreted. And then that last one is actually a serotonin receptor 6 specific agonist, that receptor that specifically localizes to the cilium. That one also was able to cause a significant change from the DYRK1A inhibitor alone. Okay, so what I've shown you today is that autism proteins converge at the cilium despite really varied annotations and known functions at the synapse and at chromatin. Makes us think a lot about how synapses and cilia are actually very similar cellular structures, really important for cell-cell communication, with very similar signaling pathways for both of them. I also gave you this idea of these chromatin regulators that also function in the tubulin code. And so that's really where my heart is right now in thinking about how these chromatin regulators are regulating both histones and tubulins. What are the post-translational changes that they're interacting on, and how does that affect brain development? Because of course the brain needs dynamic microtubules to develop typically. I also showed you that autism gene variants disrupt gut neuron migration and Xenobus tropicalis, and that acute activation of serotonergic signaling could induce evacuation. So I favor this idea that those autism genes also function in the development of the enteric nervous system, and that's why there is severe gut motility issues in that population over some of the other theories. Okay, so going forward, I think it's a really interesting yet hard thing to think about how to do is parsing the relative contribution of cilia to the symptomatology. So these proteins function both on the synapse and on the cilium. So how can we think about separating those two and thinking about what is the ciliary contribution to symptoms versus what is the synaptic contributions? Same thing for these chromatin regulators, the nuclear versus cytoplasmic functions. Like I told you, I'm excited about the chromatin regulators in the tubulin code. We can do tubulin PTM profiling now with proteomics. So we're gearing up to do some of that. And then drug screening on cilia, I think, is now possible. I mean, we're starting that work now with human cells and culture. As part of that congenital heart disease paper, we use CRISPR-I and automated image analysis in human neuroprogenitor cells to knock down various risk genes and be able in an automated fashion to quantify ciliation and cilia length. And so I think we can scale these up now, especially now that we have lines like this one, where CHD2, an autism protein, is knocked down. We see really robust phenotypes of a loss of ciliation, and the ones that do ciliate and see a little dot, they don't do it well. And so I think this is a nice drug screening platform now in cells and culture to see if we can rescue any of these cilia phenotypes. There's a few drugs that are known to affect ciliation, like lithium, but we're interested in doing a large panel of drugs to try to see what can stabilize and recover. Okay, so with that, definitely a lot of people to acknowledge. I've tried to acknowledge the students along the way, but Alina did a lot of the convergence work in the cilia project, Kate on the gut motility project, our wonderful collaborators at UCSF and beyond, our Xenopus resources, and our funding. So thank you for your attention. Happy to take any questions. Matt.
Thanks for a fantastic talk and linking this kind of very basic work to ideas about how we might be able to really, in the relatively short term at least, develop hypotheses about how to help families. One of the things I'm wondering about, so the work that you did when you, with the drug screen, what we were looking at, where was that in developmental stage that you were rescuing that phenotype?
It was acute. So it was after development was done, the enteric neurons had already come into the gut and they were already eating. And you could see the bees get in the gut, they just couldn't transit. And so it was only an acute 2-hour pulse of the SSRI, and that could move it.
And have you had any interest from any of the parent advocacy groups?
Oh, yeah.
Around potentially doing trials of SSRIs, I mean, given their safety profile and how important some of these comorbidities, including GI problems, can be.
Yeah.
And sort of, yeah. So Tell me about that.
So what I didn't mention is in these populations where you have that survey data that said, yes, GI issues, those are really severe GI issues. So these are individuals who will go 4 or 5 days without a bowel movement. They'll develop what's called megacolon. A lot of them have bowel resections. A lot of them, their caregivers are giving enemas every other night. And if you talk to these patient advocacy groups, top of the list is GI issues in terms of things that could be modified to improve quality of life. And so they are very motivated to do this. I think it's a matter of infrastructure. Obviously, a lot of these drugs are FDA approved drugs. I think the dosing might need to be different, right? My understanding, and it's embarrassing because you all know much more than I do about this, but when you give a SSRI, right, you kind of taper on slowly because there's a lot of GI stuff that's happening there to get tolerance in the gut. And I actually think you'd want to do it the opposite way with these patients, is just hit the gut hard with serotonin and then not develop tolerance to it, and then just occasionally keep hitting it to induce a bowel movement. So they're very interested in doing it. I get patient emails all the time about GI problems.
Hi, can you hear me?
Yeah.
Fabulous talk. I love how translatable it was to some of us who don't go into Frogland with you. And I'm struck by the excellent medical examples that I think are really tangible. But so many of us deal with the social and behavioral symptoms, and that's the more public view of autism, right? They're not always aware of the enema issues.
Yeah.
I just wondered if you could speak for a minute whether or not that's a space the frogs can go, or if that's a limitation of this model. If you could just speak to that, I'd love to learn more.
Yeah, great. That is a fantastic question, of course. I've kind of beat around the bush because I'm looking at other things other than the behavior, which of course is the defining feature. And so part of that is intentional. I wanted to really root this in molecular biology and looking at development of organs and tissues to be thinking about rational drug design. And so I wanted to have phenotypes that I could robustly measure and manipulate. And so some of that was intentional. There's certainly a whole body of literature in Xenopus about social behavior. Even in the tadpole stages, they can learn, they can remember, you can train them on different colors. You can make them play Frogger and like avoid these dots. Frog people are the best. So it's all possible. We haven't done any of it. I think the next thing that I would go after actually is sleep, because then you talk to families and it's gut and then it's sleep, and seizures too. But then the behavioral aspects are actually down the list for a lot of those families. Sleep, I think, is particularly tractable because they're diurnal, and you can actually watch with automated imaging analysis the circadian rhythms of the tadpoles. And again, you could do it in 96 well format, and you could drug screen on that pretty easily. But it's possible. I haven't done it. I don't have any near-term goals of doing it, but it'd be interesting to see those sorts of phenotypes. Yeah, and it's a human-specific trait, right? And so I'm nervous to look at it in the frog and sort of make extrapolate things from that. But and that's really why we focused on things like gut motility, things like that.
Great work. Thank you. I was intrigued by the comment that you made about increased dose of gene effects on cilia, that in some instances they're more or they're longer or what have you. And whether you might be able to look at the relationship between hypersensitivity to sound and specific genes.
Totally. Yeah, so a lot of the auditory mechanisms depend on cilia. Yeah, I didn't mention any of this, but most of the sensory system depends on cilia. So perception of heat, hearing, vision. The cilium is really a sensory organelle. And so in thinking about dosage effects, that particular example, they were attributing the 16p1 1.2 lengthening, duplication lengthening of cilia to being an increased amount of tau K2, which is in that locus, and they were actually just able to overexpress tau K2, and the cilia did get longer. We recently published on tau K1 loss of function, and we see a loss of cilia there too. So I think there are major players that are dosage sensitive that will affect the length of the cilium, the functioning of the cilium, whether the cilium forms at all, and could be used for those sorts of things. It would definitely be relevant to the auditory system since it depends on cilia and for a lot of the downstream signaling mechanisms. Matt.
Actually more a comment. Nikki's great question and your great answer. I do think right now we're in an environment in which sort of defining our terms and being explicit is probably more important than it has been, in the recent past. And so it's obvious in this audience that I think that what Helen has been describing is, and studying has been, large effect genes that are very often associated with severe forms of autism, which now sort of the community is calling profound autism, and distinguishing that from the broader autism spectrum, particularly out of kind of recognition of the kind of neurodivergent perspectives on ASD. And so I think that that's it's a worthwhile thing. Again, it seems like from the questions in the audience, there wasn't sort of a concern about that, but it's worth noting. And I just wanted to make the additional point, though, that the comment that Helen made about the importance of the non-behavioral phenotypes to the profound autism community is very well supported by data. It really is the case that the behavioral symptoms of autism are often very low on the list when you ask parents what their concerns are when a group has been defined based on the genetic mutation and not on the behavioral symptoms. I do want to say, though, also that with regard to the kind of the neurodiversity perspective, there is kind of a meeting of the minds there, that while the issue around kind of what it means to have these behavioral differences are a subject of a lot of discussion, the quote unquote comorbid conditions are things that in general the neurodivergent community and the profound autism community have a tendency to agree on more, like things like treating sleep abnormalities in someone who has this, et cetera. So I just wanted to point out again that it's an odd moment now that we have these political and philosophical debates within the field in the context of a much broader kind of societal debate. And so I think it's worthwhile to kind of point out where we exist on that spectrum. And we're now at 9:30. Fantastic talk. Thank you so much. Thanks everyone for being here.